Regulators of G protein signaling, or RGS, are protein structural domains that activate GT pathways for heterotrimeric G protein alpha subunits. RGS proteins are multifunctional, accelerating proteins that promote GTP hydrolysis by the alpha subunit of heterotrimeric G proteins, thereby inactivating the G protein and rapidly switching off G protein coupled receptor signaling pathways. Upon activation by receptors, G proteins exchange GDP for GTP, are released from the receptor, and dissociate into a free, active GTP-bound alpha subunit and beta-gamma dimer, both of which activate downstream effectors. The response is terminated upon GTP hydrolysis by the alpha subunit, IPR001019, which can then rebind the beta-gamma dimer, IPR001632 to IPR001770, and the receptor. RGS proteins markedly reduce the lifespan of GTP-bound alpha subunits by stabilizing the G protein transition state. Whereas receptors stimulate GTP binding, RGS proteins stimulate GTP hydrolysis. The first to be identified was SST2, supersensitivity to pheromone, in yeast, Saccharomyces cerevisiae. All RGS proteins contain an RGS box, or RGS domain, which is required for activity. Some small RGS proteins such as RGS1 and RGS4 are little more than an RGS domain, while others also contain additional domains that confer further functionality. RGS domains can be found within the same protein in combination with a variety of other domains, including DEP for membrane targeting, IPR000591, PDZ for binding to GPCRs, IPR001478, PTB for phosphoterosine binding, IPR006020, RBD for RAS binding, IPR003116, GoLoco for guanine nucleotide inhibitor activity, IPR003109, PX for phosphoinositide binding, IPR001683, PXA that is associated with PX, IPR003114, pH for phosphatidylinositol binding, IPR001849, and GGL, G protein gamma subunit. Like, for binding G protein beta subunits, IPR001770, those RGS proteins that contain GGL domains can interact with G protein beta subunits to form novel dimers that prevent G protein gamma subunit binding and G protein alpha subunit association, thereby preventing heterotrimer formation.